in this project, we're comparing the kinds of information that we get from analyzing the sequence variants in RNA-seq in addition to typical transcriptome measurements of gene expression. In particular, what we were looking at was a set of RNA-seq data sets we obtained from prostate cancer from certain subjects at UIC, and comparing these to prostate cancer data sets we obtained from dbGaP. So in addition to quantifying gene expression and doing a transcriptome similarity analysis, which I'll get to in just a minute, we also analyzed sequence variants for these RNA-seq samples. The variant calling was fairly straightforward. You just used BCF tools to call single nucleotide variants, and then annotated these with respect to where in the genome they were, population genomic databases like db-SNP nomad, cancer-related databases, as well as RNA editing from ReadyPortal. Within the sequence variants, we then followed two different strategies for prioritizing the variants. One was to assess likely germline variants to allow us to analyze admixture and ancestry for the subjects. And for this, we looked at variants that were called fairly frequently, at least 30% of the samples, had a reasonably frequent minor allele frequency, at least 1%, and had been annotated previously in db-SNP and nomad, in particular with at least a 1% allele frequency in nomad also. And then we removed any variants that were annotated in COSMIC, NCI60, or ReadyPortal, the RNA editing database. The second prioritization strategy we used was to identify variants that were likely not germline, but still somewhat common within the data set. So here we also required a call rate of 20%, a minor allele frequency greater than 1%, and then variants that were not annotated in db-SNP or nomad, so they were not previously characterized in the context of population genetics. Both of these sets of variants, we got many tens of thousands of variants, including a large number of RNA editing variants in the non-germline variety. In both cases, we ran an unsupervised admixture analysis to determine admixture groups from the variants. From the likely germline admixture, we could then correlate these with allele frequencies of nomad to make a determination of ancestry. For the non-germline admixture, we can't do an analysis like that, but we can get an idea of what types of variants are showing up and how those correlate. So the main result is this large heat map in the middle. What we see is that the USA subjects along the rows have very high correlation with specific subsets of samples from dbGaP. So here's a set right here with very high correlation. Here's another subset in the middle with medium levels of correlation. When we looked at the genetic ancestry along the left and the top here, we don't see a very strong correlation here. Most of the subjects in the UIC group are African ancestry. Most of the subjects in the dbGaP group are European ancestry, which are the purple colors. But the samples that are highly correlated have different ancestries. On the other hand, when we look at the admixture groups from the non-germline analysis, which are along the bottom and the right-hand side, we see high levels of this gold-colored group 2, both in the UIC subjects as well as in these samples that have high correlation. So here, these samples with high correlation measures are almost all characterized by this gold admixture group for the non-germline, similarly over here and in this set as well. So this was kind of interesting to look at in a little bit more detail. And what I tried to do is to analyze which variants were kind of specifically defining the different admixture groups for this analysis. And then within those variants, looking at the distribution that were either RNA editing, and I observed that almost all of the variants from this interesting group 2 were in fact RNA editing variants. We can also look at the type of nucleotide substitution that occurs, and almost all of these variants in group 2 are specifically A to I editing variants. So we're isolating a very specific type of editing event. And none of these are exonic variants. They all occur in non-coding portions of the genome. So this was kind of an interesting observation that we could identify a specific group of variants that corresponded very strongly to this transcriptional similarity. And then that gives us kind of another measure in the RNA-seq data to kind of dig into a little bit further to see what else might be going on. And I also have over here just the list of the most frequently observed genes with editing sites in this group 2 admixture. The top panel, although we didn't see a strong correlation to the admixture ancestry analysis, I did want to confirm that we're getting reasonably accurate results here. And so for the subjects from UIC, we actually also had whole exome sequencing from adjacent normal tissue. And so we were able to compare the genotype calls that we are guessing are likely germline from the RNA-seq to the actual germline variants that we can observe in the exome data set. And for the variants that are called in both data sets, a position where the genotype is callable, in almost all cases, they agree with each other. In the small cases where they don't agree, further analysis revealed that these are mostly issues of allelic imbalance in the RNA-seq. On the other hand, most of the positions that had callable variants were very different between the RNA-seq and the whole exome. Exons that were not expressed in the RNA-seq, but we could still assess variants, would only show up in the exome. And places in the region that were either not annotated exons that didn't show up in the exome panel, or intronic regions that were expressed enough to call variants only showed up in the RNA-seq subject. So if you look at the x-axis of these scale of these two plots, we see many, many more variants that just aren't comparable between the two assays. However, despite all of this, we were able to, I think, get a fair reflection of the self-reported race of the individuals. Among the UIC subjects, these were mostly Black or white, self-reported. White individuals were mostly a mixture of non-Finnish European admixture groups in the purple. The Black individuals were a majority of African ancestry, and then different levels of non-Finnish European mixed in as well. Okay, thank you.